Good morning. I'm back with part 2 of complex inheritance patterns, ways that we go beyond Mendel. And my first example is epistasis. Epistasis is a case where one gene is going to impact the effects of another gene, so has some influence over another gene. Often, one gene will hide or mask the visible output of another gene. My simple case over here on my slides is that if you have a total baldness gene, so like an inability to form hair, right, then that would be epistatic to the gene for brown hair. You wouldn't actually see brown hair because hair can't be made. And so epistasis is a way that we often will see a gene influence or mask or hide another gene and its output. I want to give you the example of Labrador Retriever. So over here you see a brown lab or a chocolate lab, and then a yellow lab and a black lab. So these are common Labrador Retriever colors, and these are highly, they've been bred, and so breeders have kept track of their genes and they know a little bit about how the genetics is passed. So I want to sort of walk you through how epistasis is working in these particular labs. I'm going to switch to a place where I can draw. So I'm switching off screen here, and here is my epistasis example, lab coat color. There are actually two genes that control how this color is going to develop in our lab. So there are two controlling genes. One of the genes is a color gene, so I'm going to say capital B for that, and this gene directs dark pigments in our Labrador Retrievers. Then we have also an expressor gene, or an expression gene that's labeled letter E often. And so this is an expression gene. My understanding is that the expression gene is a way to pull the pigment along the hair shaft so that it is sort of pulled out from skin all the way down the shaft of hair. So the expression gene is going to influence the impact of this other gene. So I'm going to say over here, this gene impacts color gene, right? The one that is above. Now, what happens to give us these three kinds of Labrador Retrievers? If you have a capital B in your genome, and you have a full expressor gene, at least one capital B and one capital E, then this particular lab will express a black coat color. The black coat color comes from that capital B black pigment being expressed, and then the expressor gene is going to pull that blackness all the way from the skin down the shaft of hair so that you get a black skin and a black coat color on your Labrador Retriever. Now, if you have small b, so you're recessive for that blackness, but you still have the expressor gene, then this gives you a chocolate lab or a brown lab. So again, we get a lighter color because we don't have that dark pigment, but we have the expression. So the brownness will be pulled all the way from the skin out and down the shaft of the hair so that we have this chocolate color all over your lab. Yellow labs are a little bit different. In yellow labs, we have a couple of possibilities. If you have capital B, right, and any other letter, but then you have a lowercase or a recessive form of the expression gene, then this will give us a yellow lab with black skin. So the black skin is from this gene over here, but the absence of this expressor gene means that the blackness isn't pulled all the way from the skin down the shaft and expressed along that sort of hair piece or follicle. So this one is going to give us a yellow lab with black skin. Or if we have lowercase b, lowercase e, we get a yellow lab with brown or light skin underneath that coat color. So this is an example of a way that this expressor gene influences how this pigment gene works in our lab color and makes lab coat color a little bit more complicated, as you will see, than what we normally find. This is the case of epistasis, so I'm going to switch back to my screen here. And epistasis is something, again, that we see, and it adds a little bit of complexity to what Mendel proposed. Linked genes are my second example for this part 2. Linked genes are the case where genes tend to be inherited as sets due to their locations on chromosomes. The closer you are stacked on a chromosome, then the less likely you are to actually cross over and complete that crossing over process in prophase 1 of meiosis. And so the first time we saw this was in things like fruit flies, where they would track wing length and eye color. Wing length and eye color, you can see this vestigial or small wings and normal wings. Eye color was quite close on a chromosome. And so it rarely gets crossed over. The closer you are, the less likely that the enzymes are going to switch you during this crossing over phase. It doesn't mean it can't happen. It just means that it happens less frequently. And so often these linked genes, they're linked, or they often get inherited as sets because they're found so close on the chromosome. We also talk about linkage to certain chromosomes. And so sex-linked traits are linked to that 23rd set of chromosomes. These are genes or traits that are located either on that X, that long 23rd chromosome, or the Y, that short sort of 23rd chromosome. Remember, genetic sex determination in humans, if you have one long and a short, we write you XY. We typically call that male. And then XX, your genetic sex would be female because you have long sort of chromosomes. In sex-linked genes, it's interesting because X and Y aren't the same length and they don't actually carry the same sequences of genes. And so this is kind of a mismatched pair that we have in our sort of karyotype that I showed you a couple lectures ago. Y is quite short. Y will only be inherited by males, so this contains things that only males genetically would get. X is quite a bit longer, and X contains some things that have to do with primary and secondary sexual characteristics, and then some things that don't, that both male and females need, things like muscle development, things like color vision, and things of this nature. So how does sex-linked traits work? Well, X-linked, if you have a Y-linked trait, you're always going to inherit that as a male, and that will be passed from sort of fathers to sons. But an X-linked trait, it's interesting that X-linked disorders are often found on the X, and they're inherited more frequently in males than in females, because males only have one chance to get that X right, or one chance to not get a mutation. So instead of having that like kind of cancer system where you have two strikes and then you sort of knock out a cell cycle regulator, males, we say, are hemizygous because they have like two different sort of chromosome possibilities on this 23rd set. And because you only have one X, you have only one chance to inherit the correct color vision or the correct muscle development or these kinds of things. And contrary to what lots of people think, this is passed from your mother. You're going to get your Y chromosome as a male from your father, for sure. So the X is actually going to come from your mother. So your mother is a carrier and then passes this on to sort of male offspring more frequently than female offspring. We see some examples like color vision, red-green colorblindness, is much more common in males. I think it's somewhere like 7 or 8% of the US population of males versus females. It's less than 0.5%. So very rarely do you see a red-green colored blindness in females, quite common in males. Hemophilia, this is a bleeding disorder. This is the Russian sort of royal family, and Queen Victoria was actually a carrier of this clotting factor problem that caused some of her offspring to bleed out and die. It was passed between the females, and then again, the males in the family got it more frequently, and they couldn't like sort of repair wounds, etc., and there was lots of deaths. And so it was one of the first well-studied examples of this because it had to do with this royal family. And muscular dystrophy is another example with muscle development, where we see muscular dystrophy more in males than we do in females. All right, why do males get X-linked disorders more often than females? I want to show you how this happens with a Punnett square. So a woman's father is colorblind, but she has normal color vision. She marries a normal vision male. What are the chances of her children being colorblind if they are males and females? So let's take a look at this really quick and look at how this happens. I'm going to switch to my drawing board over here. So if we have a genetic female, so here's our parents up here, we have a genetic female, she will be XX, and then we're going to cross her with a genetic male, XY. If we allow a capital N to be like sort of normal color vision, and then a lowercase n to be colored blindness, then what would be a female who has a father who is colored blind, but she has normal vision? What would be her genotype? Well, if she has normal vision, we know that she has one capital N that is connected to the X chromosome. And if her father is colorblind, the only X that her father can give is a colorblind X, right? So that X carrying colorblindness. If she marries a normal vision male, normal vision will be written X capital N Y. Y has no color vision genes on it, so it is left open. So let's cross these two parents over here so that we can see how this works. X, my normal vision male across the top, and then my sort of carrier female across the side. Whatever touches the box goes into the box, so X capital N, X capital N, X capital N, Y, X capital N, X small n, and X small n, Y. If we look at these children, then over here we have the females, right? And interestingly enough, because each of the females carries at least one normal color vision, then 100% of our female offspring will have normal color vision. However, over here, XYs, here's our genetic males. If we look at them, 50% of our males will be colored, sort of have normal color vision, and 50% will be colored blind. So this one, 50% of the males will have normal color vision, and 50% of the male offspring would be colored blind. So, interestingly enough, the female children of this couple cannot have colored blindness. However, the males are almost like flipping a coin. This is interesting to me because my dad is red-green colorblind, and so I am a carrier for this, and both of my sons, I only have two sons, and both of them are also colorblind. Every time I have a son, it means that it's almost like I'm flipping a coin. 50% of my sons would have like a regular normal color vision gene, and 50% would be colorblind, but in fact, both of my sons are actually colorblind. All right, so moving forward, this is showing the possibilities of like sort of different parents. So homozygous normal female and a colorblind male. You can see everybody is like normal because the mother gives that normal X to each of them. It's only in this case right here and right here where we get colored blindness. This is the most common, where the female's a carrier, marries a normal color vision male, and then 50% of her male offspring will have colored blindness. Versus this one, if you have a carrier with a sort of colored blind male, this is the case where you could get some females with colored blindness, right? So this is the only way, and even then it's like 50%. So it's still quite rare that would happen. There are lots of different tests that you can take, so you can take these online. My boys have both taken these genetic tests to see, and there's different forms of red-green colored blindness, and my kids know what they are, but you can take these tests to see if you have this as like sort of something that's problematic. All right, I want to sort of end with a couple of different ways that the environment and outside influences changes our genes and the impacts and how those genes are expressed. So I want to remind you that genes are potentials. Genes give you the potential for a certain height or a certain skin color, certain eye color, these kinds of things, hair color. But the environment can also impact how these potentials actually manifest or are shown in an individual. My first example over here, imagine leaves on a tree, where you have a tree And then the genes of each of those trees is telling a tree to make a certain shape and size of leaf, right? But when you walk around, are all leaves on the same tree identical? No, they aren't, right? Because certain trees might be smashed against a building, and so half of the leaves get more sunlight, right? Or more nutrients in some cases. Viruses can impact like how those shapes and development of that leaves go down. And so leaves in a tree, even though genetically have the same potentials inside of them, will be different shapes and sizes. Skin color is another example. You can see different people throughout the year. Their skin color is actually changing based on how much sunlight they're in and that melanin and how it's like responding to the sunlight. So skin color can change within individuals, but also within families based on sort of environmental influences. And this ultimately is why identical twins are not identical. I show a picture over here of two identical twins, but environment plays a role in like sort of what you eat, your nutrition, what kinds of carcinogens you're around. These kinds of things will impact what happens to one twin over the other. Even some behaviors that are influenced by genes will be quite different. And so twins do not look identical always, and they also don't act or behave or have all of the same mutations by the end of their lives based on what they've been exposed to and based on the environment. Last thing I want to mention are mutations. Mutations can change inheritance patterns forever if they are in germline cells. So if they are happening to sperm and egg sort of development. So this happens in errors in meiosis or damage to chromosomes that are happening in those germline cells, so in sperm and eggs, etc. Chromosomal mutations are the biggest kind of mutations that we see. These are changes that impact large sections of DNA on a chromosome, but there are some smaller ones as well, like point mutations and frame shifts. Sickle cell is an example of this, where there's a one-letter mistake, deletions, duplications, etc. that happen. And chromosomal mutations, you're deleting and duplicating, inverting, and translocating large sections of a chromosome. So I want to walk you through these types of mutations that happen that change our genome forever. So deletions are where we're going to remove a section of a chromosome. So here are some like sections labeled with letters, and D disappears, right? So that's a deletion. Duplication is where you have a section that accidentally gets maybe copied incorrectly so that there's more than one of these sections on a chromosome. So you see BC that's copied BC, etc. along our chromosomes. And duplicated, we have inversions where we actually take a section, maybe in crossing over some other mistakes, and the section gets turned around so that it is in the opposite direction. This happens a lot in certain situations, and genes often will have, they always will have kind of a starting position to them and an ending position, almost like a uppercase letter to a sentence and then a period at the end of the sentence. And so if we flip that sentence around, sometimes it becomes nonsense. It's almost like deleting something. And then finally, translocations. In crossing over, if we don't equally cross over, this is a mutation where a segment from one chromosome moves to a non-homologous chromosomes accidentally. So this is like chromosome 1, a chunk of that moving to like maybe chromosome number 2 inside of the cell, and non-equal pieces of homologous chromosomes, they're not crossed over equally. And so we get this like kind of recombining of the chromosomes, and sometimes this is problematic because it's almost, again, like you're deleting a section, or it can become nonsense where that cell doesn't work. So this is part 2 of Mendel's laws and how we break them. This has a lot to do with why we can't predict all of the traits of our offspring and design babies, right? And get all of the traits that we want in our babies. I hope you've enjoyed this. If you have any questions, join me on our Zoom meetings. Otherwise, see you soon.